Great. Thank you so much for that introduction, Ben. Yeah, for clarification, I cannot see the Twitch stream whatsoever. So people can ask questions away or say really ridiculous things or tell me my slides are ugly and I will not notice whatsoever. So go for whatever you need to in the chat. So as Ben said, I'm going to be talking about the role of dissimilatory iron reducing bacteria in organic matter remineralization and implications for banded iron formations. So I am an aquatic biogeochemist. What that means is that I'm interested in how different elements cycle in things like lakes and in oceans, how they cycle in water and how they're buried in the sediments. My primary focus for my PhD and my postdoc has been on early Earth processes. So we're talking billions of years ago, not things that I ever knew that I would be thinking about growing up whatsoever. But some really fun questions at play. And just to break down this title a little bit, dissimilatory iron reducing bacteria are referring to bacteria that reduce iron. So reduce iron from iron 3+ to iron 2+, but do not incorporate that iron into their cells. And what I'm going to be chatting about in general is experiments that I did feeding Shewanella, which is a DIR bacteria, some phytoplankton biomass, and looking at how the chemistry of these culture vessels change over time, again, with some early Earth implications. So I generally consider myself to be a geochemist, and this was kind of my foray into experimental microbiology, which is a completely different world, but incorporating still some of that geochemistry base of mine. So to provide some big picture context, in general, I know at least in the US, earth sciences is not very widely taught. So I like to start by giving some context for what I mean when I say early Earth. So shown here is a diagram of Earth from 3 billion years ago to present on the x-axis here with ocean redox state, atmospheric oxygen, and dominant photosynthesis on this y-axis. And by ocean redox state, all I'm really saying is the amount of oxygen present within any given water column in the ocean. So you can see that prior to about 2.5 billion years ago, there was really limited atmospheric oxygen. So there was no oxygen in the oceans, and anoxygenic photosynthesis dominated. We fast forward to about 500 million years ago, the world is flush with oxygen as it is now. Our oceans are now fully oxygenated, and all kinds of biological evolution has occurred in this time period. So to just give you some idea of what my interests are, I'm particularly interested within the Precambrian, which is this time period where there's no oxygen to some kind of intermediate stage of oxygen. And the reason that I find this so interesting is not just because of the shift in chemistry over time, but also because of the biological evolution that occurred during this time period as well. So we start off with the early Earth being a relatively, I guess, boring, for lack of phrasing, environment. We have some hydrothermal activity, some volcanic activity, but in terms of life, the Earth just had microbes on it that performed relatively simple metabolisms. It was void of anything multicellular. It was just a kind of bare bones Earth environment, if you will. And then we fast forward to some oxygen being present in the atmosphere and the oceans, and we start to see the evolution of multicellular life. So there's this huge transition during the Precambrian, both in terms of ocean chemistry and in terms of biosphere evolution, which at least for me makes for a really interesting time period and subject matter. And then of course, after this point, once we reach this 500 million years, that's when we get really, really rapid evolution of life. So just this part of time here is where you end up seeing evolution of fish, evolution of plants and dinosaurs and humans and all that stuff, which I weirdly care less about. So one of the ways in which we know about these evolutions, both chemical and biological throughout time, is through trace metals. So trace metals are really great indicators both for ocean chemistry through time, as well as they are necessary for the evolution of life. So I'll walk through why that is. So trace metals are great records of ocean chemistry. So again, by ocean chemistry, ocean redox, what I'm really saying is the amount of oxygen and sulfide that's present in the water column of any given ocean system. So depending on the levels of oxygen and the levels of sulfide, you end up with the burial of different trace metals. So these are things like iron, manganese, molybdenum, uranium, cadmium. Depending on the water chemistry, you'll get different abundances of these metals, different ratios of these metals, different isotope systematics, speciation, all kinds of things. So in terms of my previous research in my PhD, my focus was largely on looking at the geochemistry of these metals in modern systems. However, as a result of my PhD, I actually became a little bit more interested in the role of trace metals in microbiology and the implications for early Earth. So trace metals are necessary for all life. All microbial metabolisms require trace metals in what are called metalloproteins. So all of our cells also necessitate having trace metals. They are necessary for life to evolve. So shown here is a plastocyanin molecule, which has a copper core. So this is one of those metal proteins, metalloproteins. And plastocyanin is one of the proteins that's present in photosynthesis. So any organism that can conduct photosynthesis necessitates having plastocyanin and necessitates having copper within that metalloprotein. Well, how do we know about these records of trace metals through time? We can see them in the rocks that these oceans have left behind. So this is a figure of molar nickel to iron ratio on the y-axis and time again on the x-axis here, back 4 billion years now. And all I want you to take away from this plot is just that throughout time, Earth's history, we have records of trace metals throughout time, which can tell us something about ocean chemistry and or tell us something about the availability of these different metals for life, to diversify and for life to evolve and different metabolisms to evolve. So for example, within this plot, you can see that there's kind of a shift in the amount of variability you're seeing on that y-axis at about 2.5 billion years. And that coincides with what is referred to as the Great Oxidation Event, which is that first burst of oxygen 2.5 billion years ago. One of the main records that we have of trace metals through time are called banded iron formations. These are massive deposits that are laid down primarily during the Precambrian, so during a time in which there was limited to no oxygen. There are these massive deposits of iron oxides, so they look rusty, right? That like reddish, orangish color. They're found all over the globe. They are different sizes. They span that entire time period. And what's interesting about banded iron formations, at least for the purposes of my work, is their nature of their formation. So if we think about iron oxides in the modern world, so if we think about rust in the modern world, what we expect is that if you have an iron nail or an iron rod and it's exposed to oxygen for a while, that is how you generate rust. But as I just told you, these iron oxides and banded iron formations were developed and laid down during periods of Earth's history that didn't have oxygen present. So without oxygen present, how in the heck are we getting these massive deposits of iron that have fallen out from ancient oceans? One of the hypotheses for their formation is dependent on microbiological processes. So I'm not going to walk too into the weeds with any of these details, but what you need to get from this particular slide is that in an early Precambrian ocean where there's no oxygen present, you have phytoplankton in the surface ocean that is oxidizing iron from iron 2+ to iron 3+. This phytoplankton dies, and the iron and the phytoplankton biomass falls to the sediment. Keeping in mind that the phytoplankton is full of metals, right? Because metals are necessary for all cells and for this particular microbial metabolism to exist. So the TLDR of this slide, which I will go into some detail in, is that there are some processes that happen in these sediments that indicate that the source of the trace metals to the banded iron formations and the source of iron is actually the phytoplankton itself. So the evidence for this is that there's a similar stoichiometric ratio of trace metals between modern phytoplankton cultures and banded iron formations. So banded iron formations may as well have been found or been developed from a degradation process of phytoplankton biomass. So let me tease apart what's actually happening here in the sediments with some greater resolution. We have dissimilatory iron reducing bacteria in the sediments, which we have evidence for existing in that time due to iron isotopes. So these bacteria reduce iron from iron 3+ to iron 2+, but do not incorporate that iron into their cells. This bacteria is eating that phytoplankton biomass that has fallen through the water column. As a result of this breakdown of this organic matter, you have a release of some trace metals as well as various iron oxide species together. And with time, those metals become incorporated in the iron oxides and you get banded iron formations. So my role is breaking down just this first part of the process here. And that, again, you have these iron reducing bacteria breaking down the biomass and releasing trace metals as part of the process. But what that really means is that they're breaking down the biomass, breaking apart metalloproteins that house those trace metals and releasing those trace metals. So my role in this work was to test this process and ultimately test this fundamental hypothesis that dissimilatory iron reducing bacteria then help to facilitate the degradation of phytoplankton and the development of BIFS. So what does this actually look like in lab? Well, I synthesize ferrihydrite within lab. I grew up Shewanella onadensis MR1, which is a model dissimilatory iron reducing bacteria. So I believe this is the organism that is the second most cultured organism to E. coli in microbiological research. So it is a model DIR bacteria. And then I also was able to get photoferrotroph biomass. So this is photoferrotroph cultures that were grown up, freeze dried and rinsed and ground up. And I combined all of these different components in serum vials, which I purge of oxygen and seal from the atmosphere. And over the course of a couple of weeks, I sampled for iron and for trace metal composition. Now, because this is experimental microbiology, I also included a number of different controls. So this includes a positive control, abiotic controls, and a live control. And so these are all just vials that are used for a basis of comparison with which to compare data from my experimental vials. So I did these experiments where I fed Shewanella phytoplankton biomass. And in doing so, would hope that they would consume this biomass and release trace metals. So what I'm actually asking in terms of my research questions for this work are, one, will the Shewanella even eat this carbon? So although Shewanella is one of the most studied organisms in microbiological research, normally when it's grown, it is fed really purified forms of carbon. So things like lactate and pyruvate. It has never, to my knowledge, been fed a natural organic matter source, such as just dead biomass cells from phytoplankton. So this work in and of itself is just a test of, will they even eat this natural source of carbon? Will they be able to eat the carbon compounds that are within this biomass? And the way in which to test this would be to evaluate Shewanella's growth, which would be via measuring iron 2+ over time. The next question, assuming that they are actually eating that biomass carbon, is does the breakdown of that carbon actually release trace metals? So really what I'm asking is, does the breakdown of this carbon include the breakdown of metalloproteins and release trace metals over time? So the two measurements that I ended up taking for this 2-week period were for iron 2+ as well as for trace metal compositions. Here's what that actually looks like in the lab. So in order to measure iron, I used the ferrazine method. Ferrazine is a reagent that binds to iron 2+ and creates this really beautiful purple color, the absorbance of which can be measured on a UV-Vis spectrophotometer. And the right here are samples for trace metal measurements in a UV-Vis. So for this, I use clean sampling techniques to extract samples from my vials. I filtered these samples to measure dissolved trace metal concentrations, as well as diluted them, and did some troubleshooting, just making sure that I was getting accurate concentrations for the instrument to pick up. So these were all run on an ICP-MS. We can look at iron data first. So this is iron 2+ concentration on the y-axis here and time on the x-axis, with the biotic replicates in black and white here and the abiotic control in gray. So this abiotic control, again, is just biomass present without any Shewanella to eat it up. And you can see that there's an increase in iron within the biotic replicates that is significantly greater than our abiotic control. And this increase in iron, again, is really an indicator that the Shewanella is growing. So what this is telling me is that Shewanella is actually growing and reducing iron over time in the presence of this biomass. What this looks like in lab is a pretty stark color change. So we start off with these kinds of samples on the right or on the left here with this really beautiful red rusty iron color. And then as that iron gets oxidized, it turns this kind of mucky, I think it's technically a green color over time. So there's a really cool visual shift as this experiment was going along. So with this data alone, we can then answer our first question: can Shewanella eat carbon from that phytoplankton biomass? And all signs point to yes. We are seeing evidence that iron is increasing over time, which indicates that Shewanella is growing. If they're growing, they must be eating carbon, and the only carbon that we give them is carbon in that biomass. So then to look at this next question, right? We know that they're eating the carbon, but are they actually releasing trace metals in the process? Or in other words, we know that they're eating the carbon, but are they specifically breaking down those metalloproteins and releasing trace metals? So to do this, we evaluated trace metal composition over time. So in a hypothetical situation, if we are to assume that the breakdown of biomass liberates trace metals over time, then over time on the x-axis here, we would expect to see some change in dissolved trace metal composition. We would expect to see a change in our experimental vials that differs then from our controls, both in terms of trend and in terms of the concentrations of trace metals that we're seeing. So we would expect to see something like this, wherein we have a distinct trend and a very high trace metal composition in our experimental vials where Shewanella is fed the biomass, but yet no change, just for example, in these controls with just biomass in gray and just Shewanella present in this kind of pattern bar. So I used this kind of framework to evaluate the trace metal composition of these vials with a suite of different trace metals. So I know this is absolutely tiny here, but this includes profiles of manganese, zinc, cobalt, molybdenum, and nickel over time. And I'm not walking through all of the data at all. So just to provide you an example, this is dissolved molybdenum concentrations over the course of the experiment with the same color coordination applying here, where black is our experimental vials and these other two bars are our controls. And you can see maybe a slight increase in molybdenum over time that is well within error. And there is a corresponding increase in at least our live control here. So there's really nothing definitive that we can say here or with any other of the trace metals that implies that there is a degradation process of those metalloproteins and a release of trace metals. You're just going to have to take my word for it for all of the other metals, but there really was no distinct trend or concentration across the board when comparing our experimental data to our controls. So in revisiting this question of does this breakdown of biomass actually release trace metals, there doesn't seem to be any indication that this is the case. There's no evidence that there's any change in trace metal composition that's attributed to this process of the breakdown of biomass by Shewanella. So what this implies then is that Shewanella is eating carbon from the biomass, but it's just not accessing those metalloproteins in particular. Well, what does this mean for our overarching hypothesis? There's really good evidence still that the source of trace metals and iron in banded iron formations is from phytoplankton, as attributed to this similar trace metal stoichiometry between these two pools. But I think that the issue actually arises by thinking that this degradation process occurs specifically with dissimilatory iron reducing bacteria. And the reason why is because modern DIR bacteria do not have extracellular enzymes. So in order to break down something that is as complex as a protein, which would be represented by this polymer here, into smaller things like monomers, which would then be able to be incorporated into cells, bacteria would need to release extracellular enzymes that would help to facilitate that process. And modern dissimilatory iron-reducing bacteria are not capable of producing extracellular enzymes. There's no evidence in their genome that they are able to do so. And if they're unable to do so now, then they sure as hell are unable to do so looking back billions of years with more rudimentary microbes. So if dissimilatory iron-reducing bacteria aren't the ones that are breaking down proteins in this process, then what other heterotrophic bacteria are even there to possibly take part? Well, the other two main heterotrophic bacteria that were present during sediments at this time that we have evidence for are methanogens and fermenters. And methanogens need to take in really, really simple carbon compounds, so things like CO2. So there's no way that they really facilitated this process. But on the other hand, fermentors do in fact excrete extracellular enzymes and may have just been playing a much more critical role in this process than previously hypothesized. So if I generally were to be able to redo these experiments and had all the time and the money in the world, I would absolutely be trying the same experiment again, but using different heterotrophic bacteria to see if we saw different results to toy around with this new hypothesis. And so overall, this is just a great example of how we can look at microbiology and trace metal chemistry in tandem with one another to address hypotheses about early ocean chemistry. I found experimental microbiology to be quite difficult. As I mentioned earlier, it was my first kind of avenue into microbiology, and it required a lot of troubleshooting and a lot of methods development. So I think that I'm really curious in the future about how microbiology informs trace metal geochemistry and ocean chemistry in the early Earth, but maybe I won't be delving into the difficult parts of all of the experimental development and microbiological work. So with that, I'd like to conclude with a land acknowledgement of the Kanyan-Kehaka peoples for the land of Montreal, which is where all this work was done, and leave up just kind of a general get in touch slide. And with that, I'll take any questions.
All right. So let's go ahead and jump straight into the questions. And this is not a cutoff for questions. You're welcome to, in the chat, keep asking questions as we go. We'll basically go until there aren't any more. So the first question is, are there sources of contamination that you have to watch out for when you're measuring trace metals from long ago?
Yes and no. So in general, when you are doing any trace metal chemistry whatsoever, you need to use clean techniques. So you need to acid wash all of the glassware that you're using, acid wash plasticware that you're using, try to remove any possible residual contamination from any materials that you're using to process samples. So you are in large part accounting for any potential contamination. Also important to note is that you're using rocks from billions of years ago. So it's aggregated samples of, years and years and years, or millions of years, I guess, technically, of material. So you're looking at relatively high concentrations of these different materials. And you usually also will normalize your trace metal data with other elements that will kind of help to get at where the trace metals are coming from. So there's efforts in place in order to account for potential contamination or for kind of unaccounted for sources of metals during that process.
Yeah, that was going to be my next question was, do you have to watch out for like in the samples themselves, like where you collect them, do you have to watch out for contamination? But if you're checking them against other elements, it's unlikely, I guess, that you would have 10 different elements all of a sudden overrepresented or something like that, unless they were really present like that. Okay.
Right. And I think with early Earth samples as well, you would have to pour like a liquid concoction with a really high elevation of like molybdenum, for example, on the rock itself in order to raise levels significantly that by the time you process it, you would see really high, or unrealistically high values.
Okay.
I don't think that the general handling of a rock is actually going to contaminate it.
Okay.
The concern is much more when you actually grind down that rock and start breaking it down to release trace metals and you're diluting it. That's when it really comes into play of, we need to have this be as clean of a technique as possible to not introduce anything from the lab setting.
That makes a lot of sense. Okay. Let's see. So you had the figure where you had the bacteria with the biomass and then the controls next to it. Why do you see higher trace metals in the control Shewanella versus the control biomass?
Oh, so for this figure in
Yes.
particular.
Mm-hmm.
So the Shewanella culture inherently also has trace metals in it, because Shewanella is a microbe, so it has cells, right, full of metals.
Right.
And I was also working with really small vials and really small samples as a whole.
Okay.
So I injected Shewanella that had been grown up in a media that does have some trace metals in it. So as part of accommodating for the fact that I am introducing trace metals in that way, that is why we evaluate these concentrations by the controls. So that's what these values are saying, which tells me in this figure in particular that a lot of the molybdenum that we're seeing in our experimental vials has nothing to do with the release of molybdenum during degradation. It more so is introduced just from the Shewanella being there in the first place.
That makes a lot of sense. That's good. That's a great set of controls. That makes a lot of sense. Okay.
Yeah, we went crazy adding as many controls as possible. It is
generally
very difficult to do trace metal geochemistry in tandem with microbiology. It doesn't happen that often. And I think part of it is because you are doing things in a clean and sterile way with regards to microbiology, not wanting to contaminate your samples, but you're also looking at things from a clean and sterile way from a chemistry standpoint. And it's just very difficult. So we try to cover all of our bases by incorporating as many controls as possible.
That makes a lot of sense. But I think you did a fantastic job, because
thank you.
It makes the controls, I think, show very clearly that like you said, you're not generating any additional trace metal, and what's there you can account for readily from the control. So that's really well done.
Right. Yeah. We also I didn't include any of the data or not to it here, but we looked at the trace metal composition just of the media we were growing it in alone, just of the iron oxide that I synthesized in lab alone. We really just try to cover all of the possible bases of where are the metals in the system and how could they be contributing, so that we could really hone in on any metals that were being released as part of this process.
Yeah. Oh, that's fantastic. Let's see. Are there other DIR bacteria that could have produced the trace metals? So I think this question's asking about, like you said that the Shewanella in particular wasn't making trace metals. Is there any possibility that there was another bacteria that was produced based on the original hypothesis?
That's difficult to evaluate, mostly because Shewanella is the most studied DIR bacteria out there. And the genomic studies of various DIR bacteria are somewhat limited. So for Shewanella, there's no evidence whatsoever that they can produce those extracellular enzymes to facilitate this process.
Right.
I believe that there also is some genomic work for Geobacter, which has a lot of iron reducers in that genre as well. I don't know what the proper microbiology terms are to be fair. But there doesn't seem to be any evidence that they do any extracellular enzyme production either. So it just doesn't seem to be something that that DIR bacteria do, in which case it seems more likely that there's just another type of heterotrophic bacteria, again, that's facilitating this process.
That's really interesting. Can the bacteria use other sources of oxidizers such as nitrate or perchlorate?
So oxidize. Now I'm like going back to oil rig. That what oxidation is lost in my head, just to keep it straight. So in terms of oxidizing, other than the carbon source, is the question.
I think so. Yeah. Right.
I'm trying to visualize it to picture it in my head. I do this all the time, but it's now 10 p.m. So I have no idea.
Trust me. I understand.
It is. They are not metabolically capable of using a different kind of oxidant in the reduction of iron.
Okay.
So carbon would be their go-to, without a doubt.
Okay.
We also used, it is also worth mentioning that I used a minimal media to grow them, and that is the liquid with which all of these materials are sitting in. So that minimal media just gives the Schuanella like the bare minimum support for growth. And it does not include any extra trace metals added to it. And it doesn't include any other kind of carbon source. So really their go-to would be any carbon available, which it does seem like they will use some of the carbon available right in the biomass, but I just think that the more complicated proteins are not up their alley.
Okay. And the person that asked the question has clarified that they were referring to using a different source than the iron 3+.
Oh, not there wouldn't be anything available to them for this process now, for within this experiment.
Okay.
In terms of the early Earth, there was abundant iron oxides, which is as evident by the fact that these BIFS, right, are these massive pools of iron. So there was lots of iron coming out of hydrothermal events and lots of iron oxidation and reduction occurring. So the assumption there is then that iron is going to be the primary, is it oxidant, reductant, oxidant?
Yeah. All right.
You know, I'm just going to keep getting them confused.
The oxidizer, yeah.
It's too late. That there's lots and lots of iron available. So the assumption is that would be what they would be using.
Okay.
I don't think that there has been much other work to look at the potential for other oxidants, but I could be wrong there. That's a really good question.
Okay. Maybe it would make sense if nothing else, it would suggest that it's the dominant pathway, even if it can use other sources.
Right. And Schuanella is capable of reducing a number of other metals as well. So they're known manganese reducers, and they've been shown to reduce things like copper and uranium, I believe, are also two notable metals there. But I think that their preference, so to speak, is iron, and energetically that makes the most sense if you consider the thermodynamics of it all, which again is like out of my wheelhouse.
Sure.
But if you consider the thermodynamics of it and the redox ladder, iron, if available, is going to be their go-to.
Okay. Yeah, that would make a lot of sense then. Okay. More question. I'm trying to remember what it was. I've lost it. It's gone. So I think that's all the questions we have. If I remember my last one, I will ask it very quickly. But if not, if there's no other questions, say thank you again to Dr. Rico for a really interesting talk. It's super cool work that you're doing. It looks like you answered a difficult question too, or at least provided some of the answer to it, which is it's really neat to see how you worked from the hypothesis all the way through the experiments and to an interesting answer, even if it wasn't what was expected.
Really cool. Or what I technically hoped for.
Exactly.
Even though you shouldn't have bias, I definitely was optimistic about
it. We always have some bias. That's okay. We always have we have the answer we hope for, and that's just what we go with. But that's okay.
Yep.
Like I said, the work is super cool, and I think you did a really nice job explaining how again how you started and worked all the way through to the results that you got and interpretation of what. So,
Thank you. Well, yeah, thank you so much for the opportunity again. And I just want to make sure to welcome people to I don't know, find me online, ask questions of anything. I'm not sure who's in the audience now / would be watching this later, but if anybody generally has questions about navigating STEM, earth sciences, or interdisciplinary work, my email is always open. And if you don't end up writing this down, if you just Google Katy Rico, I'm pretty much the only person that comes up. So there you go.
Perfect. Is that your dog there?
Yes. Oh yeah. That's my dog, BB. She is fantastic, but also chaotic, as most dogs are.
Yep. I got a 2-year-old who's that explains him very well.
Yep.
That's fantastic. Well, thank you again, Katy. It's, like I said, just awesome talk. Super cool work that you're doing. Thank you again to everybody for coming out. Hope you enjoyed it. This, like I said, if you missed any of this talk, it will be uploaded to you a little bit, Twitter. Check that out, and back next week. We actually have talk from Dr. Fayer Ho, actually in the audience tonight, so she'll be presenting next week. So I encourage everybody to come back and watch that as well. In time, if I get you to hang around for just a second, Dr. Rico, we'll talk very briefly. And to everybody else, have a great weekend. Stay safe. See you next week, I hope. Good night.